So thank you, Carla, for the presentation and the invitation to speak here today. It's very difficult to follow up Dr. Aguayo now in my presentation. He mentioned a few things that I'm going to repeat right now, but it's a good thing for you. You will remember them. I don't know if it's working clearly or if I am not listening very well, but okay. Anyway, so the topic that I'm going to talk about today is about the role of glycine receptors in ethanol consumption. And as an introduction, we know that the alcohol abuse is a worldwide medical and socioeconomic burden. We know that this can affect people from all over the world, from different ages and from different economic statuses. And Chile is not far from this. You know that the World Health Organization has declared that Chile is one of the top 3 countries with most alcohol consumption in America. And also the latest report by the Chilean government by Senda in 2021 reported that more than 44% of the Chilean population drinks alcohol, or at least has drank alcohol the past month. And if we see here in the graph, they say that these percentages are been quite stable in the past years, but they have been a reduction in the scholar population. I don't know what to say about this, because this was made prior to the pandemic or right at the beginning of the pandemic, so maybe all this data may have changed by now, and also because we know what is happening, for example, in the United States, that would see that even younger people are drinking every day, right? So we know this is a very important problem. It's important for us to understand what is happening, right? And the term that is currently being used when we talk about alcohol consumption is what is called alcohol use disorder. And this is made by the NIH, AAA. And this disorder is characterized by the impaired ability to stop or control alcohol use, despite all the adverse social, occupational, and health consequences. And this can be on a spectrum of disorders. So it can be from mild, moderate, to severe. And in some cases, despite of the conditions, it can be an alcohol abuse, dependence, or finally, alcoholism. So it is important to know, for us to know what's happening, because it's a big problem, right? So what happens when we have someone that encounters an alcohol beverage for the first time? What makes this person that is going to drink for the first time to become later an alcoholic or to develop an alcohol use disorder, right? As I said, more like a general definition of addiction is something that is a loss of control over the consumption and this compulsive seek of it, right? Despite all the consequences. But in a more biological definition, my biological terms, we can say that the process of addiction is defined as a form of neuronal plasticity that is induced by the drug, right? And this can involve both synaptic or structural changes in specific areas of the brain, right? So we know that this is what happens right here at the end. We already know that our brain is different, right, after we have become an addict. But what happens in the middle is still rather unknown. We know that throughout this process there are episodes of dependence when we need to drink alcohol. There are some other cases when there's a withdrawal. We stop drinking. And fortunately for some people, they can recover and they don't become an alcoholic, right? But in some of them, there are relapses and they drink again. So as for the person that asked before, what causes this, or what leads to some people to recover and to some people to relapse, is multifactorial, right? It can be genetic, environmental, social factors. So it's really, that's why there's this huge question mark at the middle. We still don't know. So the important thing is to try to understand when or where these plastic changes that I just spoke. So where are these plastic changes occurring? These are occurring in specific parts of the brain, something that Professor Aguayo already mentioned, but I'm going to expand a little bit in there. And the main regions where drugs or alcohol is affecting is called the reward system. And this is neuronal circuitry that some of the most important regions are the VTA, the ventral tegmental area, the nucleus accumbens, and the prefrontal cortex. And this reward system is very intricate and is very connected to also the basal ganglia, something that Professor already said as well. For example, from the VTA, we know that they're going to release dopamine to the nucleus accumbens, and the VTA is very next to the substantia nigra. And the substantia nigra also releases dopamine, but now to the striatum. And we know that dopamine is very regulated in the brain. Only few areas release dopamine. So when there's an impairment here, or for example, the degeneration of the neurons of the substantia nigra can cause Parkinson's, right? So we know that when dopamine is affected, it's gonna cause some problems. And this reward system, as has been mentioned, in the evolutionary sense, it was meant to incentive rewarding stimuli, natural stimuli. They were important for survival and to maintain the species. And what is that? For example, finding food, water, and sexual activity for reproduction, right? So currently, we don't really need to have an incentive to tell us that we need to eat or we need to have water. Like, we know that, right? It's very available for us. So right now, this system is used by other type of rewarding stimuli. For example, drug of abuse. And the literature actually refers that drugs hijack the system because they get into the system to make you more incentive to continue to look for it, to go for it, and to consume it and do it again in a more quantity. So this reward system is very conserved in many species, and I'm going to change now to the mouse model. And as we can see here, in a mouse brain, we have the same regions that I just mentioned you, so that makes us able for us to study the system in a mouse model. As we can see here is still the VTA, the nucleus accumbens, and the prefrontal cortex. What's important is that the nucleus accumbens is like an integrative nuclei. It receives inputs no other from the VTA and the prefrontal cortex, but as well from the hippocampus, the amygdala, the hypothalamus, etc. So it needs to incorporate all the signals to finally determine whether or not you are going to go for this drug or for something that is pleasure for you. For example, I'm not very keen for chocolate, but I do like fries. So that's gonna depend on whether what you like, right? And something that also mentioned Dr. Aguayo is that the drug of abuse are going to cause, this massive release of dopamine from the VTA towards the nucleus accumbens. And this is something that has been known for many years. If you know, this study was made in the 1980s. And they showed that it doesn't matter, regardless the mechanism of action of any drug, they're always going to cause a massive release of dopamine into the nucleus accumbens. For example, after administering amphetamine towards freely moving rats, you can see over 1,000% more dopamine levels in the nucleus accumbens, and this will last for at least 5 hours. Also for cocaine and for ethanol, obviously with different concentrations, not as high as amphetamine, but still a large increase in the levels of dopamine in these regions. So any kind of drug is going to produce that, is going to activate this reward system. So if we go back to our model here, we know that the nucleus accumbens is receiving all these signals when you consume a drug, or for example, when you drink alcohol. So this nucleus accumbens is going to receive all these excitatory signals. They're going to make that this nucleus is activated or is overactivated. So in a way, it needs to have some sort of a mechanism to maintain a balance. Like Professor said, we need to have like a balance in excitation and inhibition. So what would happen in this nuclei then? Something that he also mentioned is that this nuclei is entirely inhibitory. More than 95% of all the neurons in the nucleus accumbens are GABAergic, are going to release GABA. And they also have interneurons that are GABAergic as well. So But besides that, we think that after getting dopamine from the VTA and glutamate from all these other regions, maybe they are also expressing other inhibitory receptors, such as the glycine receptor. So now I'm going to talk about a little bit about glycine receptors, very briefly, because I know that you've been hearing a lot about it, I'm sure. So we know that these are inhibitory ligand-guided ion channels. They are widely expressed in spinal cord and in brainstem, but just a couple of years ago, we now know that these glycine receptors are also expressed in upper brain regions, some of them part of the reward system, and specifically in the nucleus accumbens. We know that they have like this pentameric structure, like we see here in the cartoon. And they can be homomeric with just alpha subunits or heteromeric with alpha and beta. And what's important to remind you is that the expression of alpha 1, alpha 2, and beta in the nucleus accumbens, that was already reported. So by having this in mind, we thought, okay, will this glycine receptor have a role now in the regulation of the nucleus accumbens or in ethanol consumption, right? But before we get to answer that question, we also need to remind that glycine receptors can be modulated by ethanol, right? This is like the proposed mechanism of how ethanol is acting on glycine receptors, is that glycine receptors, when are activated by glycine, they're gonna open, the pore that is gonna lead to the entry of chloride inside the neurons, which is gonna cause an hyperpolarization. And now, in the presence of ethanol, ethanol is gonna act indirectly through the dimer beta gamma. Ethanol is gonna cause more availability of beta gamma. So now beta gamma can act or can bind to basic residues that are in the intracellular domain. And what is this going to cause is that the more chloride is going to enter the neuron. So we say that this is the potentiation of the inhibitory current, right? And these basic residues, they are in the intracellular domain, were initially studied for the alpha-1 subunit, and there are two lysines, the 385, 386. And how can we see this in electrophysiological recordings? We see this as, for example, this atypical glycine current. And when we perfuse the cells or neurons with glycine plus ethanol, we see a potentiation of the current, right? We see that the amplitude of the current is bigger. Okay, so this is what I just said, that it has been studied for the alpha-1 subunit. So what happens for the alpha-2? We know that the alpha-2 also have the requirements, the molecular requirements to bind beta-gamma. But in these studies that they were made by Dr. Yevenes, he saw that only the alpha-1 subunit, the homomeric alpha-1 subunit, is the one that is capable of being potentiated by ethanol, but not alpha-2. And as we see here in the graph more clearly, even at higher concentrations, alpha-2 is not potentiated, right? So this was around 2010. Years later, we found out that there's something important about the beta subunit for the alpha-2 receptor. As I mentioned, these are, the lysines, they are in the intracellular domain for the alpha-1, both homomeric and heteromeric. But for alpha-2, now it's a lysine and an arginine, the 389 and 390. So what happens now when we have this heteromeric formation of the glycine receptor? What we see here is that now this insensitive receptor, this alpha-2 homomeric, now becomes sensitive when we have this heteromeric conformation. This is what we can see here in the graph. Even alpha-1 homomeric, the potentiation with 100 millimolar goes no longer above 50%. But with the alpha-2 beta, the potentiation is almost higher than 80%. So making this conformation, alpha-2 beta, now the most sensitive to ethanol. Okay? So if we know that this now heteromeric receptor is able to potentiate by ethanol, what happens if we mutate these residues? that we know that were important for alpha-1, right? So that we have this lysine and this arginine, and now we're going to mutate them to two alanines. So what's going to happen now? We lose the potentiation that we saw with the native receptor. This is what we see here in the graph. The most sensitive conformation, alpha-2 beta, with a high potentiation, is completely blocked when you mutate these basic residues in the intracellular domain. So for this receptor, alpha-2, to be able to be potentiated by ethanol, needs both the beta subunit and the presence of these basic residues in the intracellular domain. So far, we're going to do a little recap so we can understand the next experiment. So for now, we have been saying that right in the reward system, when we drink alcohol, we increase the levels of dopamine in the nucleus accumbens, and there's this activation or overactivation of the nucleus accumbens. But we also know that there's the expression of different subunits of glycine receptors in the nucleus accumbens, and especially that the conformation, alpha-2 beta, is the most sensitive to ethanol. So now we can go back to the first question. Will this subunit, especially the alpha-2 beta, will have a role in the regulation of the nucleus accumbens and then in ethanol consumption? So to test this, we developed a mouse model that is called the knock-in alpha-2 mouse. And what this mouse has is that it has a mutation, this mutation that I just showed you. The lysine and the arginine are mutated by two alanines in every alpha-2 receptor in this mouse. So this is a global mouse. So every glycine receptor in his central nervous system is going to have this mutation, right? And they have like a C57 background. So what happens with this animal? The glycine receptors of this animal act just as a normal, or sorry, a native receptor. They both can be activated by glycine and increase the permeability to chloride. However, now when there's the presence of ethanol, only the wild-type animal, which expresses the wild-type receptors, are going to be able to be potentiated by ethanol, but not the knock-in ones, right? That's the only main difference. Both of them can be activated, but only one of them can be potentiated, okay? So the first thing that we wanted to test is what would happen in the nucleus accumbens. We know that the alpha-2 subunit is expressed in the nucleus accumbens, so will this have a role? Will they have a difference in this region? Is that in this case, what we did was electrophysiological analysis in dissociated neurons from the nucleus accumbens, and this is what happens with the wild-type animal. We have here a glycine current activated by 50 micromolar, and glycine plus ethanol, a high concentration, we see this huge potentiation of the current, right? This is in the wild-type animal. What happens now in the knock-in? We see this. A complete or almost complete reduction of this potentiation that we observe in the wild-type animal. When we now see here in the graph, we see that in all the concentration that we tested, increasing concentrations, we see the same response. There is a significant reduction of the potentiation that we saw in the wild-type animal. So in a way, we're saying that these neurons are losing some inhibition, right? We could say that. So if we think that maybe these animals are losing inhibition, what would happen with the excitability of these neurons? So the next thing that we went to test was what would happen with the firing of action potentials. Okay? So in the wild-type animals, in controlled conditions, we see that if we stimulate the neurons with a current, we would find a certain number of action potentials. Now, in the presence of ethanol, again with a high concentration, we see that this number is reduced, right? Nothing much happened after we perfuse the brain slices. These are in brain slices with ethanol and strychnine. But the main thing is this, right? Ethanol is a depressant neuron, so we know that it's gonna lower the activity of the neurons. So this is what normally happens in wild-type animals. What happens now in the knock-in alpha-2? Nothing. Ethanol does nothing, and the same number of action potentials remains the same. This is what we see more clearly here in the graph. This is what normally happens in wild-type animals. In the presence of ethanol, around 35% of the number of action potentials is reduced. This is what normally happens. It is what should happen, you know, because ethanol is a depressant drug. But in case of these animals that they have these glycine receptors that are mutated and they are not able to be modulated by ethanol, we don't see a difference in here. So we know that something is happening in the nucleus accumbens. We know that these neurons are losing inhibition, right? They should be inhibited, but they are not. So if the nucleus accumbens remains active, we think and we propose, okay, if the nucleus accumbens remains active, then something must be happening in the behavior of this mice. So then we went to test what would happen when we test them for alcohol consumption. But first, what happens if we test them for administration of ethanol, right? Before the test that I'm going to show you, we did many controls to see how the basal motor control or a normal activation of the mouse before we went to the ethanol test, and they were completely normal and they act similar to wild-type animals. So then we proceeded and we went to see what happens when we administer ethanol to these mice. So the first test that I'm going to show you is called the fixed-speed rotor test. And this test is to test the motor activity of animals. And as we see here in this model, mice are placed in a rotating rod that is going to rotate at a fixed speed. And this is no problem for mice. They are very capable, right? So the first 3 days of the test, they stay here and they have no problem to maintain themselves the 120 seconds that the test lasts. There's no problem for them, right? But what happens when we inject them now in an IP injection of 2 grams per kilograms per weight? This is a high concentration, right? This is an intoxicating concentration of ethanol. And what happens is that 15 minutes post this injection, what happened with them? They fall immediately, obviously because they are under the influence of alcohol, so they fall. As we can see here, what we're measuring here is the latency to fall. So if we see here the black curve, which shows the wild-type animals, we see that the wild-type animals require more than 90 minutes to finally be able to get to the 120 seconds above the rotor rod, right? However, the knocking alpha-2 mouse, they do it around 75 minutes. So it seems to be like they are recovering faster from this injection of a high concentration of ethanol. And to rule out that this might be something involving the metabolism of ethanol, what we did was to measure the blood ethanol concentration in these genotypes 1 hour after the injection of these 2 grams per kilograms. We see that there is no difference between the two genotypes. So these changes that we're seeing here are not because the knocking animals are metabolizing ethanol faster, right? And the second test that I want to show you about the same regard is called the loss of writing reflex, or LOR. In this case, mice are injected, again, an IP injection, but in this time, an even higher concentration. This is a 3.5 grams per kilogram. And this is a sedative concentration. So mice are sedated, and they are placed in a supine position, as we see here, supine, with lying in their bags. This is not normal for a mouse, right? So the mouse are sedated, and so we measure the time in which the mouse now right themselves. That is the name of the test. When they right themselves and they put in the normal prone position. And again, we see that the knocking alpha-2 animals, they take around 25 minutes, more or less, to be fully awake and now in the normal right position. But wild-type animals require more than 35. So again, these animals seems to be recovering faster for this injection of ethanol. So this is administration of ethanol. Now, what we really want to know what would happen when they would consume. What happens with voluntarily drinking. So for that, well, again, we also measured the blood ethanol concentration, and we saw there was no difference between them. Okay, and so to measure ethanol consumption, we use this test that is called drinking in the dark because, as Professor said, it's performed in the dark phase of the light and dark cycle of mouse because it's the cycle where they are more active. And in this test that lasts 4 days, the first 3 days they are able to drink a solution of 50% ethanol for 2 hours. And then on the 4th day, they can drink for 4 hours, right? In this case, we change the bottles. We take the bottle of water and then we put the bottle with ethanol. And what we can see here, if we check as well, first the black curve, which is the wild-type animals, we can see the wild-type animals start drinking relatively low concentration of ethanol, and they increase day by day the consumption, right, in a more escalated way. However, knocking alpha-2 animals, they start drinking a higher concentration, and they remain rather stable up to the 4th day when they can drink for the twice as time, so the consumption is even higher, right? So what's interesting here is that we did some measures as well, blood ethanol concentration after the 1st day of consumption and the 4th day of consumption. And the difference that we see here, we also see it in this part. We can see that the knocking alpha-2 animals reach levels of around 120 milligrams per deciliter of ethanol in blood, which is a lot. And the wild-type animals, they no longer pass around 70. I marked this dotted line around the 50 milligrams per deciliter because this is the level that in most countries is what is determined as the limit of when a person is drunk, right? If you're driving and a policeman stops you, and if the alco test or the alcoholemia test, you will get more than 80 milligrams per deciliter, then you will be detained, right? So just to make the comparison to humans. But what's also interesting is that this pattern of consumption is called binge drinking. I don't know if you know about the term binge drinking, but binge is when you do something rather quickly and a lot. If you're that kind of person that, for example, watches TV shows or stream shows, for example, one season or the entire series in one sitting, that is called binge watching, right? You do all that immediately. In drinking, it's the same. It's binge drinking. Is when people drink a lot in a very short period of time. The NIH defines binge drinking as when people can drink and achieve levels of above 80 milligrams per deciliter in less than 2 hours. For people, there's around 4 drinks for male and 3 drinks for female, right? In average, obviously. What we see here is that these animals appear to have this behavior, this binge drinking, because in a short period of time, in less than 2 hours, they reach levels very past the 80 milligrams per deciliter, right? So these animals may have this binge drinking behavior, which is something that is very popular nowadays with teenagers or people around the world. So it's a very complicated and it's not the best way to start drinking, right? So anyway, these animals, they seem to have this pattern of drinking, and they drink a lot more than the wild-type animals. And after the 4th day, even though these values were not significant, the knocking alpha-2 animals, they do drink more than the wild-type animals. So this was just the last thing that I wanted to show you. Just now as concluding remarks, we can say, you know, from what we said at the beginning, is that the nucleus accumbens is the key region that processes these rewarding stimuli, in this case as well, drug of abuse, and how this nuclei is overly activated when we use a drug of abuse. And for that, the expression of the glycine receptors in this nuclei can be helpful, right? And how the alpha-2 beta conformation might be as an important target for ethanol action, since this is the most sensitive conformation of glycine receptors. And finally, that we can say that the alpha-2 beta has a role in the regulation of the nucleus accumbens and both sedation and ethanol consumption. So To finalize what we propose that it might be happening in this case, that we have here like a typical neuron from the nucleus accumbens and MSN, that when we drink alcohol, it will get activated, right? It will get all these inputs, all these excitatory signals, and also dopamine from the VTA. So these neurons will be overactivated. So in case of wild-type animals, that they had these native receptors, they can be potentiated by ethanol. We would sort of have like this balance between the excitation and the inhibition, right? So what happens now when we have this animal that it has this mutation is that it no longer can be potentiated, and we saw that they were losing some of the inhibition with the electrophysiological recordings. So we can say that these neurons might be not balanced in terms of overexcitation. So we can say that these neurons remain active, and so for the whole reward system remain active, and this animal drink more ethanol, right? So that's what I wanted to show you today. I'm gonna thank, of course, the Laboratory of Neurophysiology in the University of Concepcion, our team ethanol right over there. Our PI, of course, Dr. Lisa Aguayo, and Dr. Greg Homanix from the University of Pittsburgh for the generation of the knocking alpha-2 mouse, and of course our funding for the NIH project. So thank you. Yeah. Yeah, that is something that we actually want to know. This is we do it like this because this is how the protocol is, right? We're using a protocol that is widely used in literature, and so what we're trying to do right now is that using this sort of new tech with licometers that would make you how many times the mouse access to the bottle, how many times is actually drinking. I don't know if I can say right now, because this is something that we're trying to develop right now. So, but at least I think that they must do it like through times, like right at the beginning, then they stop, then they go back again. It's not like they drink the first hour and they stop. It's like that they're moving around during the time. But that is something that is very interesting and that we would like to know as well more clearly. Sí. Sí, aunque no lo creas, sí. La mutación lo que hace es que el receptor ya no pueda potenciarse. No, el ratón está modificado genéticamente. El ratón está modificado genéticamente. Se generó un genotipo especial de ratón que tuviera esa mutación en todos los receptores de su organismo. Entonces, el ratón es así. Entonces, lo que nosotros estamos probando ahora es que si esa mutación, como es específicamente en una subunidad del receptor de glicina, podía tener algún efecto en el comportamiento del animal, y eso es lo que vemos. No es que está producida por el alcohol. Lo que estamos viendo es que el alcohol no está teniendo efectos porque ese receptor ahora está mutado. Esa es la diferencia. Es algo que se podría llegar a saber, o sea, como le preguntaron también al profesor, no sé si fuiste tú mismo, claramente hay mutaciones que pueden estar ocurriendo en las diferentes proteínas que van a estar regulando en el cerebro, entonces quizás esta es una de ellas. Nosotros no lo sabemos, estamos proponiendo que quizás el receptor de glicina podría ser uno de los tantos proteínas que están involucrados en la regulación. Entonces quizás hay gente que sí tiene mutaciones en esta proteína y que hace que sean más predispuestos a consumir. Claro, no sabemos exactamente si que hay personas que lo tienen en este momento. Puede que sí, puede que no, pero por lo menos en el modelo animal vemos que solamente teniendo la mutación ya los hace más predispuestos a consumir. Quizás sabemos varios que tenemos. No sabemos. Lo que les mostré aquí era solamente en machos, pero también tenemos los estudios hembras. No tengo el gráfico en este momento, pero una de las razones por las que quisimos separar es porque hay una diferencia en consumo entre macho y hembra. De hecho, se sabe que las hembras consumen mucho más que los machos ratones. Ratones. Ah. No, mira, según las encuestas de acá de Chile dicen que las mujeres consumen menos, pero eso cada vez se va equiparando mucho más. Pero en este caso nosotros hicimos la separación porque es muy difícil hacer estudios en hembras dependiendo del ciclo estral que tienen. Entonces, dependiendo del ciclo estral, pueden haber días que pueden consumir más, que pueden consumir menos, entonces es muy difícil hacerlos coincidir. ¿Ya? Entonces, por eso tendemos a separar los estudios. Lo que yo les mostré solamente alcohol, pero en hembras pasa absolutamente lo mismo, solo que es en concentraciones mayores. Yes, that is something that it was surprising for us because we were expecting that since alpha-1 is the one that is most expressed in the brainstem, we know that they have altered the Lord. So we thought that maybe there was not going to be any effect with the knock-in alpha-2. But what we saw was that there are many levels of how awareness and activation are controlled, not only in the brainstem, but there are also reticular formation, for example, in the midbrain, that we know there are neurons that release glycine from there. So maybe they are the expression of glycine receptors in those regions, and also in the cortical regions, because be aware, or for example, when you're under a surgery and you're going to be sedated, you don't want to remember what happens, you don't want to feel anything, and you just want to wake up from it. So there must be a lot of levels in which sedation is induced. So we think that maybe the alpha-2 are not the ones in the lower part of the central nervous system, but in the upper brain region. So they might be some other areas that are also expressed in this subunit that we were not aware of. I'm sorry, I didn't get. Yeah, because the basic residues for the basic nature are more able to bind beta gamma. Yeah. Mm-hmm. Yeah, no, not in more general terms. I mean, like I said before, we did the test for ethanol. We performed several other, for example, for grip tests, if they had food class reflexes, or how the weight of the animal during development, the size of the brain. We analyzed all those things prior doing all the experiments, and for example, doing, I don't know, open field, rotor, all of them, and they were all completely similar to the wild type animals. There was no like a striking feature that we would say, okay, there's something different, something wrong with this animal. Absolutely not. That is quite different, for example, with the knockouts alpha-2. That's when we saw some differences. But with the knock-in, since it's a mutation that is only affecting the activation by beta gamma, and the receptor can be activated by glycine, we didn't observe any changes in them. Yeah, we haven't tested, but we would say that no, because this mutation is especially specific for ethanol modulation. So we wouldn't expect to be changing other. I mean, we don't know, maybe, but we haven't tested. For example, it's different from what Professor Aguayo showed earlier, because it was an aging animal, and it was an animal that had other mutations, not specifically in the glycine receptors. So that what we saw over there is that there were changes in ethanol, in sucrose, but in this type, since it's just the mutation for the modulation of ethanol, I would say that it's more specific to that. Because also this type of mutation has been tested prior, for example, in HEX cells for other modulators, for example, propofol, and propofol would still be potentiated. It was not affected by that mutation. So that makes me think that if we test other things, other drugs or other components, for example, sucrose, it would not be affected. Podría ser, es que el tratamiento para el etanol es bien complejo, en el sentido de que se utilizan fármacos que no son específicos para evitar que tú consumas más, sino que, por ejemplo, hay un medicamento que hace que los efectos adversos de alcohol sean más fuertes, por lo tanto tú te sientas tan mal que digas, ok, no quiero consumir más. O también mucho trabajo grupal con psicólogos, psiquiatras, entonces no hay como un tratamiento que apunte específicamente como para decir estás dejando de consumir. Por lo tanto, puede ser de que los tratamientos sí involucren o afecten el consumo de alguna otra sustancia, porque claro, estás cambiando de alguna forma los mecanismos de regulación del cerebro, entonces prácticamente puedes estar afectando otra activación también. Gustavo? Ah. Many. He's the expert on that. No, yeah, of course. I mean, it took about 10 years later to finally realize that. So obviously it was something that it was very in mind to say that alpha 2 was not sensitive to ethanol. So, well, most of those studies were made by Braulio, so you can ask him as well. So the thing is that what they claim is that now this beta subunit is adding some more stability to the glycine receptor in terms of increasing, for example, the open probability when the channel is being activated. So it's more about a conformational change that would stabilize the opening of the pore. So now the ethanol can be potentiate the receptor. I don't have much more answer for that. It's mainly what is expressed that it would give some sort of a conformational change that now would allow the receptor to remain open longer than just the alpha 2 by itself. That's all I can say. Mm-hmm. Mm-hmm. No, the homomeric alpha 1 is higher than the heteromeric. It's what? Is higher than the heteromeric, yes. So beta is doing different things, opposite things. So with alpha 1 is actually lowering the sensitivity, but with alpha 2 is increasing it. Yeah. Not me specifically, but the prior works said that yes, that they can both bind beta gamma. They made some GST pull down, and they both bind to alpha 1 and to alpha 2. But just that they needed this extra modulation, for example, that could be given the beta subunit to now be modulated by ethanol. But both of them can bind, physically bind to beta gamma. Right? Other subunits, you mean? The other subunits models of other subunits? Yeah, I mean, Yeah, what I just saw you, that it was mainly the experiments that I made was for the alpha 2 knock-in, but there are also studies with the alpha 1, and it happens pretty much the same. They also have a higher consumption. They are also have these changes, for example, in LOR, because the alpha 1 is more expressed in spinal cord and in brainstem, but mainly they have like the same results, but at a different level. For example, we saw that for alpha 1 they consume higher than the knock-in alpha 2, but it's basically the same response for them. Of what? I mean, it could be because I mean, I think it has to be some sort of difference because of what is the subunit that is most expressed in the nucleus accumbens. We know that the alpha 1 is more expressed than the alpha 2, but they're both there, and they're in both type of neurons, both the D1 and the D2. So both of them can be regulating what's happening in terms of alcohol consumption. So I don't know. For some cases, some people might say that the alpha 1 would be the most important, but we see here that the alpha 2 is also having a role. So I think this is as a generalization, we can say that just glycine receptors, not just a specific subunit, but the ones that are expressed in the nucleus accumbens. Okay. That is just like I said, like today it's not much. People are not talking much about alcohol abuse or addiction. It's more like about this alcohol use disorder. So it's more like someone that has a more severe disorder than someone that has a mild disorder. So if you have, well, I guess that no, of course. What they say today in the NIH is just adding some more features for saying, for example, if you are in a severe part of the spectrum, if you I don't know, like you have been put your life in danger of other people, or I don't know, like adding more characteristic that would probably lead you to the side of actually saying, okay, this is an addict person, this is an alcoholic, right? So it's just adding more features to it. Yeah. What is your spectrum? get it. I mean, yes, that's what I've been telling is that glycine probably is not the only one that is involved in the regulation. So are many things. For example, the people from Asia, you know that they have this mutation in an enzyme that metabolize ethanol, and they can get all these bad effects of ethanol more than other people. So they have this sort of mutation probably in other parts of people are not from Scandinavia. They can drink more and more, and they probably don't feel anything. So maybe glycine receptor is something that is mutated or is different in some of them, and that is something that, like I said, these are all studies that we made a couple of years ago, but it's all very new still. So these are all made as well with acute alcohol. We don't know as well what would happen with more chronic. So like I said, yes, it could be something that it would be interesting, for example, do these GWAS genomic studies to see that maybe all these people that they are alcoholic, they may have a mutation in the glycine receptor. So yeah, could be good to know.